Dr. Mason is an associate professor at Florida State University. Olivia got her PhD at Oregon State, and then she went on to do a postdoc at Caltech, which is where we met. We were lab mates in the orphan lab, and we've been friends and colleagues ever since then. So it's great to have her here today. And Olivia went on to do a postdoc at Lawrence Berkeley, and then she started as a professor at Florida State. And it's great to have you here, Olivia. We'll be doing a
Yes,
follow-up to this. Great.
thank you. So you can all hear me all right and see my screen?
Yep, go
Yeah?
straight.
Okay, great. Yeah, I was so excited to give this talk and to see Jake and see Lauren again, actually. So she graduated from Florida State a little while ago, and we don't really get to see each other very much. So it was nice to be able to reconnect and look at this data that she generated from her time participating in the course. All right, so I'm part 2 of this talk where we're looking at oxygen as a driver of microbial community structure and diversity in the Northern Benguela upwelling system. So as Jake said, I'm Olivia Mason. I'm an associate professor at Florida State University. We're in Tallahassee, Florida, which is in Florida in the Southeast United States. Here's my contact information and also my website for my lab. All right, so Lauren already did a great job going over our keywords. So we use a lot of what people refer to, you say, as jargon when we talk about microbial ecology. So I hope that everything's clear and people are following along. One thing that I did want to point out is that when I say microbe, I mean bacteria and archaea. All right, so this is an image from microbial ecology of expanding oxygen minimum zones. And so we've talked a little bit now today. Lauren introduced her work that she did for her PhD here in the Gulf of Mexico in the dead zone that that happens every summer. But we'll focus more now on the work that she did when she was in Namibia participating in the course. So if we just sort of step back for a minute and consider the global ocean, Lauren's already defined these, but dead zones or hypoxic zones are areas with dissolved oxygen concentrations below 2 mg per liter or 62.5 micromoles per kilogram. Dead zones, I'm sorry, are low in dissolved oxygen, but they're also enriched in the byproducts of microbes, right? So these are microaerophilic and anaerobic microbes that produce these byproducts that include carbon dioxide, methane, nitrous oxide, and hydrogen sulfide. So they're not only low in dissolved oxygen, but they're enriched in these other species of chemicals. All right, so we have very visual evidence of the stress that low dissolved oxygen places on larger organisms in these environments. So dead zones are inhospitable to macrofauna and megafauna. And so this is actually what we refer to here as a crab jubilee. I don't know if that's just an American expression, but when you have low oxygen conditions where organisms cannot obtain sufficient oxygen to carry out their metabolism, they will actually flee the water column. And so you'll find that there are crabs on shore, and even fish sometimes participate in these, jubilees where they go to an environment where they can obtain oxygen, right? And so often that's just going onshore or near to onshore. So these are from, this is a crab and fish jubilee that was happened off the Alabama coast. This is actually a different situation though. These are dead crabs. So this is off the coast of Oregon here in the United States. And in this environment, there's an expanding oxygen minimum zone. So it's lasting longer and it's getting larger. And so what happens in that dead zone or that OMZ is that hydrogen sulfide fluxes from the sediment to the water column and actually kills these crabs. And so they wash up onshore. And there are some birds that are taking advantage of these, these organisms that were unable to obtain sufficient oxygen or were, killed by hydrogen sulfide. And so they wash up onshore and these birds are feasting on those animals. So Lauren presented this work today, and so I'll just put this here as a visual to show how impactful river input is into the Gulf of Mexico. But again, just sort of stepping back and thinking about just deoxygenation of the global ocean, that is one of the primary consequences of global climate change. So warming waters are expected to enhance stratification and decrease oxygen concentrations globally. OMZs are predicted to increase their expanse and severity, and a lot of attention has been directed towards coastal hypoxic zones, or dead zones as we call them. So this is a really different type of dead zone, though, than what Lauren studied, than what we're going to look at with the Northern Benguela upwelling system, because this is a nutrient driven, as Lauren said, this river drains a large part of the continental United States. And you can see where you have this fresh water that is nutrient rich, particle rich, meeting the Gulf of Mexico, meeting the ocean. And so this fresh water is overlaying on this high salinity Gulf of Mexico seawater, and it stays trapped at the bottom. And you have this nutrient, these input nutrients fueling a phytoplankton bloom. Those cells die, and heterotrophic microbes consume that biomass, driving the oxygen down. And you have long-term stratification until there's intense enough storms to mix that water column, so you have low oxygen. Now, this is kind of cheating, because this isn't really what we're talking about, because this is actually a red tide. So this is off of the coast of Florida. And so it's cheating because I'm using it as a really good visual, right? Where you have nutrient input and you have this massive phytoplankton bloom. So this is going to, these cells are going to die, and they're going to, be decomposed by microbes that are going to drive the oxygen down. But this has the extra problem, which is that often these phytoplankton blooms can be toxic. So we have this as a problem every summer off the coast of Florida, where we have nutrient input drives phytoplankton blooms, and we have low oxygen, but we also have sometimes toxic blooms forming off the coast of Florida. All right, so if we again just think more globally of what happens in oxygen minimum zones, one manifestation is basically the energy is shunted away from larger organisms towards microbes, right? So as you're looking at, when you have oxic conditions, you have a large portion of the energy going to mobile predators. But as you move across that oxygen gradient, you have less and less energy going to mobile predators and more energy going to microorganisms. And I would just note that when we talk about hypoxia, and we can see, visually we can see when we look at something like the crab jubilee, that larger organisms that can move will move to seek oxygen, because it's not sustainable in that low oxygen environment. But microbes are able to carry out even aerobic metabolisms when you have very low oxygen concentrations. But if we look at this, this figure from the Wright et al. Paper, we can see that as you go from an oxic environment and you move deeper into the water column, where you're encountering low oxygen conditions, that you actually have new niche space open up, right? So you have new microbes that are able to carry out their metabolism in this low oxygen environment. And when you get low enough, you can actually have methanogenesis in the water column, which is going to produce methane. But this stands for terminal electron acceptors. And you can see that in the oxic environment, this is the primary electron acceptor. And as you move into low oxygen conditions, you get a diversity of terminal electron acceptors. And you can also run these reactions in reverse. And so we're going to look at this in reverse a little bit today. And this just is, I like this illustration, although it's not up here in the dysoxic to suboxic water column. This is on a particle in an oxic environment, but the same microbes would appear here on this particle, where you have decreasing oxygen concentrations as you move into the interior of this particle that's falling to the seafloor, and where it will remain for thousands of years. So you have a diversity of metabolisms that are coming online in this small particle. And so we're going to talk a little bit about sulfur oxidizers. And Lauren already started talking about ammonia oxidizers. So these are two metabolisms that are going to be one of our focus today. All right, so she showed this map and she showed our oxygen concentrations as you move from the surface deeper into the water column. So let's revisit our primary question here. How does dissolved oxygen concentration impact microbial diversity and community structure in the NBUS? And Lauren already showed her, how she collected samples, and she ended up getting 46 samples. And as she said, we carried out I-tag sequencing of 16S rRNA genes. And she did a nice job explaining why we use 16S rRNA genes. It's because it's an excellent taxonomic marker. So, we're always jealous of people that can collect their samples and use a microscope and identify the organisms in their samples and then can count them to get at their abundance. But we cannot do that with microbes, and I refer to bacteria and archaea. So we have to use molecular techniques, and we use this, universal gene to look at the taxonomy of the microbes in our samples, but we also can get at their abundance. And so I think Jake actually presented some work using I-tag sequencing already. And Lauren already talked about this, but we are just pulling out a small piece of this 16S gene. And this is a good region to sequence. It gives you a lot of information about the microbes in your community. And then we just sequence these amplicons on a MiSeq. So this lets us determine microbial community structure and diversity. Lauren presented our RGNO data at the phylum level. So what I decided to do was to present at the species level. And so she already showed that you see a difference in terms of the abundance of the different phylums, across an oxygen gradient. And so what I'm looking at now, as I said, is species level. And so if we just look at diversity, so diversity is both richness, which is the number of different species that you have, and evenness. So what is the abundance of those different microbes in your samples? And so one thing that we see here is that when we just look at diversity, we have a lower diversity in our oxic samples than we do in our dysoxic samples, which might be surprising as you think about how important oxygen is. But let's go back to this for just a minute. As we go from our oxic samples deeper into the water column, we see that we have new niche space opening up, right, where you have different terminal electron acceptors or electron donors that can be used. So we have an expansion of niche space here. And so this is why you see a greater diversity with your dysoxic samples. And then if we continue just looking at diversity, we see that our suboxic samples have the lowest diversity. So if we compare just looking at the number of different species, we can see that we have greater richness of microbial species, again, in these dysoxic samples. If we look at evenness, we can see that actually our oxic samples are fairly similar, or very similar, in terms of how abundant our microbes are compared to dysoxic. But then we see that there's a very large difference between that and our suboxic sample. So Lauren already showed these types of ordinations. So what we're trying to do here, a lot of people have a hard time following non-metric multidimensional scaling ordinations because you don't have defined axes. But this is a non-parametric way to look at this multivariate data. So, just think about what we're talking about here. We have thousands of microbial species in our samples. So how do you take that multivariate data and compress it down onto just a couple of axes to capture the differences in your microbial communities depending on different environmental variables such as oxygen concentration? So this is really one way to do that. And so one thing I would point out is that when you, each one of these points, regardless of the shape or color, represents a sample that was collected from the water column. So if you have a sample that's, let's use these as an example, you have a sample that's here, comparing that to a sample that's here, these microbial communities are really different. So the more similar the samples, the closer they would plot together, the more similar the microbial communities. All right, so one of the things you want to know is you want to look at the beta dispersion across your samples. So If your beta dispersion is, your dispersion around the mean, if it's not significantly different, then you're justified to go on and look to see whether or not your clusters are significant. So I carried out Permanova analysis, and I determined that in fact, these different clusters, so these are suboxic, dysoxic, and oxic, are significantly different. So that means that your clusters are significant. So that is helping you understand that oxygen is certainly one of the primary drivers that's structuring these microbial communities. So now the next step is to overlay your environmental data onto this, and you can see that in this vector is indicating that oxygen is increasing. And so how you can interpret this is that as you go along primarily the second axis, your samples that are here, and then as you go along this axis, your oxygen concentrations increase, and you have your highest oxygen concentrations here. So oxygen is certainly one of the primary drivers structuring these communities, as well as depth, salinity, and temperature. So we're starting to understand what the microbial community looks like in the northern big whale upwelling system, and in particular, what factors are really driving the distribution of those communities. All right, so we know we have significant clusters, but what microbes are leading to the differences in these suboxic, dysoxic, and oxic clusters? So I then carried out a simple analysis, which just tells you the average percent contribution of individual variables or species that contributes most to the dissimilarity between objects or samples in this dissimilarity matrix. So what microbes are really contributing to the differences that you see in these clusters? That's the next thing that you want to determine. So if you, so that simple analysis revealed that there were several hundred microbes that were really contributing to differences in those samples and how they clustered. And then you want to know if those abundances are significantly different across your oxygen gradient. So this is a heat map where you have higher abundances when you get to yellow, lowest abundances in blue, and these samples are sorted just like Lauren did. These are sorted by low oxygen to high oxygen, higher oxygen. So suboxic, dysoxic, and oxic. And I'm sorry, I know this is a hideous figure. Each one of these, what you cannot read is a microbial species. So we're going to take a closer look at this. But one thing I really wanted you to look at here was this sort of block of samples of microbes, right? So across an oxygen gradient, you can see that you have higher abundances when you're looking at the suboxic samples, and then the abundances decrease as you see increasing oxygen. So let's take a closer look at some of these because you can't read that. So what I did is I looked at the most abundant microbes, and these most abundant microbes were making up a large percent of those that we just looked at on the heat map. And so what we see as being abundant is called SUP05. All right, and so this is a type of microbe that we're going to learn about today, and this is it has highest abundances. So the bigger the circle, the greater the abundance of a microbe. Its greatest abundances are where you have suboxic to dysoxic conditions. It's quite abundant actually in the water column in general, but it does decrease in abundance. The other one that we're going to look at is this nitrosopumelus that Lauren already talked about as being the most abundant microbe in the Gulf of Mexico dead zone. So let's take a closer look at these. All right, so SUP05 is really abundant, but most abundant where you have suboxic conditions. Nitrosopumelus is most abundant where you have suboxic to dysoxic, and that abundance decreases where you have increasing oxygen. And I just would say, we're going to talk in more detail, but both of these microbes are aerobes, right? So I mentioned that microbes are perfectly capable of carrying out aerobic metabolism even with low oxygen conditions. So these are some of the most abundant microbes in the NBUS water column. So we're talking about SUP05, which is a bacteria, and Nitrosopumelus, ammonia oxidizing archaea that Lauren already talked about. Both are significantly inversely correlated with dissolved oxygen. If we plot these and let's just look at the abundance across that oxygen gradient, you can see that SUP05 is actually always very abundant, as I said, but its abundance increases with decreasing oxygen concentrations and reaches a maximum in these suboxic samples. Nitrous apumilis, this archaea, is not as abundant as SUP05, but it shows the same trend, right? Where you have lower abundances with oxic conditions, and then that abundance increases as you go across that oxygen gradient. And so what this is showing is the same ordination, but the bubble size indicates how abundant these two really important microbes, clades of microbes, are SUP05 and this nitrous apumilis or ammonia oxidizing archaea. So I hope you can see that when you have suboxic to dysoxic conditions, that these SUP05 type of bacteria are most abundant, and the same for these ammonia oxidizing archaea. So why does this matter? So the first part is, well, let's look at and see how dissolved oxygen influences microbial diversity and community structure. And now we've narrowed this down to being to actually just looking at the specific microbes that are most abundant, and also their abundance changes across that oxygen gradient. So I'm not sure if any of you have heard about how challenging it is to grow microbes in the lab. I think that we were talking about this with Jake's talk when he was presenting his work about growing a microbe in the lab. So it's very challenging for a variety of reasons, but we only can grow about 1% of all microbes in the lab to study their physiology. But this is a rare instance where we're actually, Bob Morris's group was able to isolate and characterize a representative of this SUP05 bacteria that's so abundant in the NBUS water column. So this shows the cultured Candidatus thioglobus singularis. It also is showing evidence for sulfur globules. They were able to obtain this culture from Puget Sound and from the DCM and the North Pacific Gyre. So what is this SUP05 doing and why is it important? Well, it's a type of gamma proteobacteria that oxidizes sulfur, and it's also capable of carbon fixation. All right, so we know already that SUP05 is one of the most abundant, if not the most abundant microbes in OMZs. So we already have this information that they're really important in OMZs. And in our NBUS samples, we would agree with that, right? Our analysis revealed that SUP05 was up to 40% of the microbial community in some samples, particularly the suboxic water masses. So that's what this is showing here. So this is relative proportions, and you can see that SUP05 abundance decreases with increasing oxygen, but that it's always very abundant. So why does that matter? Well, the first step is that it's a sulfur oxidizer. So we've all probably seen these figures and are familiar with this very much with this group, where there's evidence of sulfur plumes that you can see from space. So why does that matter? Well, we know that hydrogen sulfide is toxic, and we've already seen pictures today in my talk of dead crabs that washed up on shore because of hydrogen sulfide. And so what is going to convert that hydrogen sulfide to sulfur, where you can see that from space, like what's shown in this paper? Well, it could just be abiotic, but it also could be a biological, the result of a biological metabolism, such as something like SUP05. All right, so actually, Lavick et al. Attributed the detoxification of sulfidic waters that we know leads to these massive fish kills off the African coast to SUP05, right? So it's a sulfur oxidizer, and it is abundant enough to be able to detoxify that water column. So that's really important. But it doesn't stop there. Why else is SUP05 really important? So Bob's group was able to culture another SUP05. This is Candidatus thioglobus autotrophicus. And it's different than the first isolate because it's a facultative anaerobe, right? It's still a sulfur oxidizer. It's still a chemolithoautotroph, but it produces nitrite, which is going to accumulate in the water column. And so it's important that it's a facultative anaerobe because not only can it use oxygen when there's sufficient oxygen, but it can switch its metabolism and actually use nitrate as a terminal electron acceptor. Well, it can't carry out complete denitrification, which is really significant because that means when you have incomplete denitrification, you're likely to see N2O being evolved. And so this is the genome of this newly cultured microbe. And these are genes that are involved in nitrogen metabolism and in particular nitrate reduction. And so they actually showed that this new SUP05 does evolve N2O from incomplete denitrification. So this is really important because not only can SUP05 detoxify the water column, but if there's not enough oxygen, it can switch to denitrification, but it doesn't carry out complete denitrification, which means that it's probably evolving N2O in the water column in the N-bus. All right, so Lauren already did a great job talking about why nitrosuppumylus, or this ammonia oxidizing archaea, is important particularly in terms of nitrogen cycling in OMZs. And there's a lot of evidence that has shown that this type of organism, this type of archaea, is really important in OMZs. So in particular, Stewart et al. Showed that it's actually a dominant energy source in the Eastern Tropical South Pacific OMZ, and Peng also showed that there was active ammonia oxidation and nitrification that corresponded with AOA in the ETNP. So it's really an important group of microbes. But also in the N-bus. So Fussell and Frame both reported that there was active ammonia oxidation in the water column in the N-bus. So we know that there's a lot of evidence that shows that AOA, these ammonia oxidizing archaea, they're aerobes, that they're really abundant in low oxygen marine environments. And so that includes in our work, which Lauren showed in the Gulf of Mexico, where these AOA can be up to 40% of the microbial community in some hypoxic samples. But let's look at the N-bus. So Lau et al. Had already reported that AOA were up to 33% of the microbial community in some of their low oxygen samples. And we also found that there was an enrichment of AOA in the N-bus with this archaea comprising close to 20% of the microbial community, with the highest abundances in dysoxic and suboxic samples. So this is a plot of their relative proportions, and we can see that their abundances are highest with suboxic and dysoxic when conditions are suboxic or dysoxic, and decrease with increasing oxygen. Okay, so why does this matter? Again, what are the ecological implications of having high abundances of this ammonia oxidizing archaea? Well, we know that AOA can actually dominate N2O production in some regions of the global ocean. Santoro et al. Reported that ammonia oxidizing archaea may be largely responsible for the oceanic N2 source, including in OMZs. So, did we answer our primary question? How does dissolved oxygen concentration impact microbial diversity and community structure in the N-bus? And I hope I've convinced you that DO concentrations are one of the primary drivers that structures microbial communities and diversity in the N-bus. So why is this significant that we have high abundances of this type of bacteria, SUP05, and this type of archaea, these ammonia oxidizing archaea, when DO concentrations are low? Well, we know that SUP05 can detoxify the water column, but both can operate, SUP05 can operate as an aerobe, and nitrosuppumylus is an aerobe. So they're going to continue to draw down oxygen where you already have low oxygen conditions with their metabolism. But probably the most important is that SUP05 can switch to using nitrate as a terminal electron acceptor, and both are a known source of N2O. And so this means that we have these really abundant microbes that are potentially producing N2O in the water column. So that potent greenhouse gas. So that's a summary of what we learned about the microbes in the N-bus. So I'll stop there and invite any discussion that we might have.